let's talk now about how eukaryotes reproduce. Now eukaryotes, unlike prokaryotes, actually have to go through mitosis, or they go through the mitotic phase. Since we just talked about the cell cycle, we now understand that most of the cell cycle is actually interphase. It's the preparation for mitosis. Mitosis is only a very small part of the replication process of a cell. So now specifically we're going to talk about the mitotic phase. Let's just talk about mitosis and the steps in mitosis. It happens fairly quickly for some cells. Other cells might take a little bit of time. So the mitotic phase is a period of cell division following the replication of the chromosomes that ensures a copy of each chromosome is distributed to the daughter cells. So the daughter cells are the new identical cells, identical to the original cell. So mitosis creates two identical daughter cells. Keep that in mind because that's going to be important from here on in. Now the mitotic phase involves a few different things. First is the division of the nucleus. And the functions of mitosis basically are growth, regeneration, and repair of tissue. So if you get hurt, your cells go through mitosis to fix that. And asexual reproduction. And I know we've said that eukaryotes are sexually reproducers, which is true. However, the cell itself, because it's creating an identical cell, is actually asexual reproduction. Also during cytokinesis, which is a step right at the end of mitosis, we're going to see the division of the cytoplasm. This occurs along with telophase, which is a phase of mitosis. And cytokinesis actually differs for animal and plant cells. And again, we're going to talk about that a little bit later. Now, we've already discussed interphase here, and interphase is about 85 to 90% of a cell's life. So much of a cell goes through interphase. And this is a picture of a stained onion root tip that shows you interphase. If you notice, here's the cell. Here's the plant cell. We have all of our DNA right in here, and we have our nucleus. So this is actually perfect. This is a perfect picture of interphase. Now let's talk about the steps of mitosis, and we're going to go into each one. First we have prophase. So prophase is the beginning of it. Then we have metaphase. Then we have anaphase. Then we have telophase. Now you can actually spell this out with PMAT. So PMAT, prophase, metaphase, anaphase, telophase. However, if you have something else to help you remember, please do so. PMAT, that is the step-by-step process of mitosis. So let's first talk about prophase. Now in prophase, we're going to see the chromatin coils, or the chromatin coiling to form chromosomes. Remember, the chromatin is the visible form it's the bundled form of DNA. Well, actually, we now get to see the chromosomes. So here in this picture, here are the chromosomes. The nucleoli will disappear. We haven't really talked about that, but just know the nucleoli disappears. The centrioles will begin to move to opposite ends. And the centrioles are, if you think about it, they kind of act like sticky tack. They're important for the cell division process. So just know that these are going to go to opposite ends. And the nuclear envelope begins to break down, so that basically the membrane of the nucleus is going to break down, so the nucleus goes away for a little while. And if we were taking a look at that stained onion root tip, we would see this. This is prophase. Notice that it's starting to kind of unravel. You can start to see some of the chromosomes in here, and this is what we would actually see if we were looking at it under a microscope. Now metaphase is the second phase. Metaphase, in this particular place, we have chromosomes moving to the center of the cell. So remember, in prophase, the chromatin uncoils, basically, or the chromatin becomes visible to form chromosomes. In this phase, those chromosomes are going to line up into the center of the cell, which is called the metaphase plate. So think of meta being middle. Think of the M, meta, middle. So all the chromosomes line up directly in the middle of the cell. In a metaphase, this is probably the easiest one to see under a microscope. You'll see all the chromosomes lined up directly in the middle of the cell. Now the third step is the anaphase. Now anaphase is the part where the spindle fibers begin to pull each half of the copied chromosomes to the opposite ends of the cell. So remember when we talked about chromosomes, how we have one chromosome actually has two parts to it. It has one side the chromatid and another side a chromatid. So they're stuck together and they're called sister chromatids. They are exactly the same. The DNA is the same, the chromosomes, the genes, everything's the same on them. So now in anaphase, the spindle fibers, which is sort of like, if you think of a spider's web, is pulling apart those sister chromatids. So now we have a set of identical chromosomes on each side of the cell. So here are your spindle fibers. That's like your sticky tack. And then we have some of those, the spider webs pulling the sides of the chromosomes apart. Ana means apart. So again, you have that A. Ana means apart. Anaphase. And in anaphase, again, it's probably one of the easiest to see because you're starting to see those chromosomes on the opposite ends of the cell. So even this one over here, you can start to see is the beginning of anaphase. This one right in the middle is pretty much close to being the end of anaphase. And the last stage of mitosis is called telophase. And in telophase, the cell membrane begins to pinch in half. So notice in the picture, we sort of have this pinching in the middle. And the cytoplasm is actually divided. Now this is called cytokinesis. It happens at the same time of telophase, but cytokinesis is different for plants and animal cells, and we're going to talk about that in the next couple slides. The spindle fibers will begin to disappear, so those spiderweb looking things are actually going to go away. The nuclear envelope will begin to reform around this, so it's going to begin to reform around those chromosomes. The nucleoli will reappear, and the chromosomes will uncoil, and basically they're going to go back to that chromatin, which is that bundle of DNA that's inside the nucleus. So telophase, sometimes it is hard to understand and see what telophase really looks like under a microscope because it might look a little bit like interphase. Now this one up here is interphase. Notice the difference between this guy and this guy. You can see what's known as a cell plate because onions are plants. So we're going to talk about the cell plate here in a second. But notice that each side we have the chromatin curling back up. The nucleus is going to sort of become whole again. The nuclear membrane will form. And now we're going to have two cells. So here's just another picture of what it might look like. We have interphase, which is kind of normal, so we're going to have our DNA replicating in this part. Then mitosis, we have prophase, metaphase, anaphase, and late anaphase. Sometimes they distinguish the two, but for the end of course test, they will not. And telophase. Now, cytokinesis occurs separately, or I should say occurs at the same time as telophase. However, it looks different for animals and plants. So for animals, in this picture you can sort of see a little pinching. It's called the cleavage furrow. So right here, you're going to see this cleavage furrow where the cell is pinching off. And this creates a kind of a cytoplasm on this side and this side. So basically the cytoplasm is replicating. So this way we have a nice clean break between the two sides. So in animal cells, they basically just create a pinching off. They create a cleavage furrow in order for them to separate. In plants, however, they actually have to rebuild the cell wall because plants have cell walls. So what happens here is that a cell plate is created. And you can sort of see it in this picture here. So this divided line right here, that's replicating the cell plate. And the vesicles of the cell will actually form that. The cell plate kind of becomes a little bit whole, and then eventually it becomes the cell wall once it's actually finished. So in this SEM picture up here, or I'm sorry, the TEM picture up here, you can sort of see these vesicles creating a cell wall or a cell plate, and it will eventually become a cell wall. So in this picture, which you've already seen before, you can kind of see the cell plate right here. It hasn't become a full cell wall yet, but it's close enough. And remember, just to wrap things up, the first stage of mitosis is prophase. Then we have metaphase, anaphase, telophase, PMAT. Alrighty, PMAT.